Okay. All right. Bye-bye and see you. Okay, so this is a part 2 of my lecture. Ala and Natalia, you missed the past one, the part 1, but that's okay. And in fact, I have a PowerPoint in my Google Classroom, so you may want to take a look if you're interested in the part 1. Okay, so part 2, I will be talking about enzyme technology for the synthesis of oligosaccharides. This is basically a research that I've been doing in my laboratory. Oh, I have to turn here. Okay, I mentioned in the beginning of my class that my research, the research in my laboratory, two things. Okay, of course, you always like to say you want to do something good for mankind. It just makes you feel better. But basically, I have two parts: the effective treatment and prevention. Last time, the first time I was talking about the effective treatment, which is a target-based therapy, recombinant human antibody for therapeutic and diagnostic. So target-based, you probably have heard, which means that you can treat the disease right on target. This will reduce the side effect and make the treatment more efficient, okay? And in my own laboratory, I use antibody as a magic bullet to bind to the specific source of the disease and try to cure the disease, okay? And was dealing with antibody and also about the diagnostic part. Now, the other side of my research is related to prevention, and that I have been using enzyme technology to produce oligosaccharides using the raw materials from agricultural waste in Thailand. And as Monica was talking last time, she was talking about insects, and insects are rich in chitin as well. So that could be related to my talk as well. All right, so today I'm going to talk about this part of my research. This part of research is, in fact, I have been working closely with Professor Haltridge. Okay, he's my husband too, but I worked with him before we married, actually. Okay, I've been working with him. He's the main person. And also Professor Vincent Ising in Norway. He is helping a lot with the enzyme chitinase and chitosanase. So, so that this is, other people are mainly dealing with antibody engineering, but these are the two people that I've been working with on this research that I will be talking to you, okay? So I mentioned in my first lecture last time that the research in my laboratory is dealing with a technique that I was very happy that in 2018 received a Nobel Prize in Physiology and Medicine. No, this is not, it's not 2011, it's 2016, okay, I think it's a mistake. So the Nobel Prize were given to, sorry, oh, this is the wrong slide, it's not this one, okay. The Nobel Prize in chemistry is given to 3 scientists, okay. This is about the molecular evolution. And on the first part, I was talking about these 2 people who are dealing with phage display technology. Okay, that both these 2 got an other half of the Nobel Prize. Frances Arnold, okay, she also received half of the Nobel Prize, and her work is related to the directed evolution of enzymes. And today I will be talking about the research by Frances Arnold. Okay, so let's hear about the modern evolution theory. You probably know about the evolution that, okay, let me turn, okay, sorry. Let me change something a little bit here. Okay, so you probably have heard about, you know about evolution. Okay, our planet start 5 billion years ago from the Big Bang. And of course, and during this part the billions year, there was a lot an evolution starting from carbon, nitrogen, oxygen. They hit each other to form RNA, and then start to form cells. And then in just the last millions years ago, 100 millions years ago, then the animals is show up in the history of the planet. And when the scientists study this evolution, they come up with a pretty simple theory called evolution theory, that basically all this evolution that we see from the beginning are composed of only 3 things. Okay, these are the mathematicians who study evolution. First, there's a mutation, the recombination, and then the selection for the best fit. And we know that these mutation mean there's a gene. So DNA is a very, very important basis information of life. And the DNA can mutate. And the mutation of DNA lead to the different protein, okay? And also, not only the mutation, but there must be a recombination as well. And then there is a selection for the best fit. Okay, so, and that was in nature. It takes billions of years to get anything evolved that you want. But what Frances Arnold did, that she received a Nobel Prize, is that she forced the evolution to happen in a test tube. And that's why it's called directed evolution. That means for the selection, you can try to force the selection so that you can get the evolution to fit what you want. So if you want certain kind of selection, like this, for example, color code, blue, red, and green. So if you force that selection for whatever you want, and you subject that DNA into this round of mutation and recombination, you will get the protein that evolve in your direction. So this is the basis of the directed evolution. Okay. Okay, yeah, this is my, so you can select, so that is a basis of directed evolution. Now, okay, from this is just to summarize again, that if this is occur in nature, it would take millions years to get evolution that you want. But for the directed evolution, we can do this in a test tube. Again, we follow the nature, meaning that we have a select set of genes. We create mutagenesis to have a collection of genes with different mutation. And then you do a recombination, mainly combining these mutations together, and then do the selection, then screen for whatever we want. So you have a library of protein from the genes that have different function, and then you do the selection, okay, and to get the good one, and then subject to several rounds, just like this modern evolution technology. So this is the basis of directed evolution. So basically you can get the enzyme that you like by the selection. All right, so my laboratory, so this is the explanation from the website of the Nobel Prize Foundation. So they talk of, is what I just said before. You have a gene of enzymes, you create mutation, okay, it produces enzymes of many kinds with different mutation, and you select for the one that has a property that you like. Keep it several rounds of selection until you get the bad one is discard, the good one is keep, and finally you get an enzyme that is improved. So maybe this looks very simple to you, but it happens to be very powerful, such that a person who discovered this got a Nobel Prize, okay, which is Frances Arnold, a female scientist, as you see. Okay, so my laboratory, I've been working with phage display, but because it's used the same concept of molecular evolution. So 20 some years ago, I decided to work on this as well. But before you do this, first you need to have an enzymes. Okay, this slide is to show you in detail when you talk about mutation. All right, and then you do the recombination. The recombination can be done by you had a set of different genes with different mutation. You cut this gene into small pieces, and then you recombine together. And then you have a library of recombinant genes, and then start to do the selection and keep several rounds, okay? The techniques of this molecular evolution is basically many kinds of molecular biology techniques. You have to deal with mutagenesis or error-prone PCR, chemical treatment, DNA shuffling, okay, the screening. And also there's a high technology, use the robot to select in a droplet of water or something. There's so many kind of techniques available. When I first started my laboratory 20 years ago, in Thailand, we don't have that much money. My lab is very small, so I started some more simple technique, which just using a select screening on a plate, which I will show you later. And I decided to use DNA shuffling technique. It was very powerful then. So the idea of DNA shuffling is that you have a set of genes with recombination, you cut them into small pieces, and you shuffle them together like this, and then you can have a gene with the different mutation, combination of different mutations, and you hope that certain combination of mutation generate an enzyme that has a better property than the original enzymes. All right, but as I said before, the screening process is important because you have a gene, but the gene has to produce the enzymes. So you have to have an expression system to produce that gene. And I've been working with enzymes, and this is an enzyme from bacteria. So it's easier to do engineering. So I start with the enzyme from the bacteria. And of course, if you want to express enzyme, you need a simple enzyme expression system. And then I use enzyme, of course, we use E. coli, Escherichia coli is a very simple, the most studied bacteria to study gene expression. All right, so 20 years ago when I start my laboratory, so I come up with our own expression vector to express enzyme. All right, but if you study, I don't know if you have some background in molecular biology, maybe a little bit, you would know that if you want to express recombinant protein in E. coli, you need to put the gene of the enzyme that you want to express into this expression vector. So you would clone into MCS or multiple cloning sites. Once you clone the gene of the enzyme in, then you can put this into the bacteria and let the bacteria produce the enzyme for you. And now, to put the gene in, you have to make sure that the gene is under the control of a promoter. But if you know the gene expression, the gene can only express if that is under the control of the promoter. And that promoter will be the site where the RNA polymerase bind, and then it starts to read the gene, the DNA into the RNA before it gets translated by ribosome to make a protein. Okay, so the vector of the vector, one thing to be considered that is important is that the promoter. So one of the two promoter that I use a lot in my laboratory is very useful is TAC promoter and T7 promoters. These promoters are based, the T7 promoter belong to this PET expression system that I think is owned by Thermo Fisher. A TAC promoter is a common promoter, right? And then in this case, when I engineer the vector, I put histidine at the end of the enzymes. In this case, it's 10 histidine, but sometimes it's 6 histidine. This is depend on different scientists. These, in addition to 10 histidine, I also have this epitope. This is called MYC epitope. It is a short amino acid with this LDYKDDDK. It is a short peptide sequence that can bind, but there is an antibody that can bind very specific to this amino acid sequence. This tag, extra tag on the enzyme is useful for purification of the enzymes, okay? Now, when we talk about the directed evolution of the enzymes, When I first started the laboratory, that was, okay, you can see we published this work in 2008. So that's quite a long while ago. So the first thing we have to do is we have to find a system to express the enzymes. And in this case, because it is an enzyme amylase, this enzyme can cleave starch. All right, it can cleave starch. And what we did is that we have to develop a method to screen for the enzyme. And as you remember, I said before that when you do directed evolution, you have to have the way to generate a mutation in the gene, and you have to have a way to isolate the enzyme that have good properties. So in this case, I would put the genes that produce enzyme amylase, okay, and then put in the E. coli. And if E. coli can secrete the enzyme out, it will have a clear zone. These clear zones means that the enzyme can degrade starch. And if you remember, starch can be stained with iodine and it becomes black, okay? But if the enzymes is produced from the E. coli, and then the enzymes can degrade starch. So there is no starch, so they cannot stain with the iodine, so it will become clear. So in this case, you can look at the size of the clear zone to determine which enzyme has a good property, a better property than the original enzyme. So you would look for the clear zone that is bigger, suggesting that this enzyme is secreted more or is more active. But remember, we did the directed evolution techniques. So first you have to have a gene of the enzymes and you would digest them. Okay, so you would digest this gene and then you will combine them together and then clone back into the gene. So it would be a library of the gene with different mutation. And one you just screen the enzyme, first screen you do on the plate, and then you have to move to do the secondary screening on ELISA plate. And you can see that in the well that has a very yellow, it means they could degrade all the starch so they become clear. So you can pick that enzymes and look for the mutation, all right? So this is one kind of experiment that we can do to improve the enzyme amylase. But in that time, unfortunately, we were not able to find any enzymes. It is better than the original enzyme from Bacillus, hydrolyte Bacillus, but it is not better than the enzyme already sold by the big company. I was only the owner of a small laboratory. So we didn't do anything more with this enzyme. The student who did this received a PhD though, because this is a very difficult, not easy technique. So it's enough for the student to get a PhD, but it's not enough to publish or sell this enzyme, okay? Later on, they would be related to the substrate that Monica was talking last time. I also interested in enzyme chitinase, okay? Chitin is the shell of many insects, including crab, prawn, all kind of insects. As Monica has mentioned, it is a skeleton, is very, very strong. It is the second most abundant cellulose carbohydrate, biopolymer, second to cellulose. You know, carbohydrate is made of sugar, right? The largest, the most abundant biopolymer carbohydrate. Biopolymer is cellulose, making trees, plants, right? And the second largest, more abundant polymer is chitin. All right. So in Thailand, we also have a lot of chitin from prawn and insects, shrimp, crab shells. So I was working on chitinase since 2000 something. All right. So another student of mine, which is she's a master's student, did the same thing. She would clone the gene of enzyme chitinase and degrade into small pieces, all right, and then shuffle them back together to get this gene. Okay, so basically you have a genes, you create mutation, and you have different genes. you cut them into small pieces, and this would look like in this gel, and then select only the part that are equal size about this one, and then recombine together. This part would be equal to this part, and then get back a library of a gene of different mutation, and after that put into the E. coli and do a very similar screening, just like with enzyme amylase that degrades starch. This enzyme chitinase will degrade chitin. So if you have an agar plate with chitin, all right, and if your E. coli can produce and secrete the enzyme chitinase that can degrade chitin, you will see a clear zone. All right, and you can look for the clear zone that is larger. It means the enzyme is either secrete more or more active. And after that, you do a secondary screening on ELISA plate, just like I showed you before. This time, we were able to obtain only one enzymes, okay, that is better, show better activity than the original enzyme, okay? When you want to study enzymes, okay, this is when I was collaborating a lot with Professor Haltrich. He is a food chemist, and he's working on enzyme, and he's an enzyme expert. I'm not an enzyme expert, so I've been asking him to help me with the enzyme analysis. So when you want to compare the enzyme, you have to look at the kinetics value, like Km Vmax or Kcat, all right? So basically, I don't know if you get to learn enzymology, but I will skip that part. But if you look at the Kcat over Km, okay, the higher the Kcat over Km, it means it is the better the enzymes, all right? So these are the two gene that is the original enzymes, that gene of the enzyme that I did mutation, and these are the two mutation. This is the mutation that my master students select, and that shows some improvement. Okay, it's only four improvement from the original enzymes at pH 3, and about twofold improvement at pH 6. Okay, even though it's only four improvement, this is good enough for commercialization. If you can make everything faster than for 4 times, it could be useful for make it cheaper if you want to use the enzyme industry. All right, so we have this enzyme, and we are just about to try to use this enzyme to produce something useful, which I will tell you later. This work was published in 2009, and then just 2 years ago, I get a chance to collaborate with Vincent Ising from Norway. If I show you in my map, I got to know him, and he is a real expert on chitin assay enzyme. He is an expert on enzyme acting on carbohydrate in general, and one of the carbohydrate he interested in is chitin. All right, he did some using a high expensive machine like NMR, HPLC. Then using this technique, he was able to confirm that the enzyme that is improved, or A3, this enzyme that's improved, is faster. The kinetic is faster than the original enzyme. So the conclusion is, it is possible to apply directed evolution techniques to improve the enzymes. And this enzyme can be useful in the synthesis of oligosaccharides that I will show you later. All right, so the first conclusion is that first you have to have a expression system to produce the enzymes, so that you can screen for the enzyme with the improved activity. And in my laboratory, we use an E. coli expression system, because E. coli is a bacteria that is easy to study. Everybody knows about this enzyme. All right, so we use this E. coli. But next, so we have 2 enzymes with the PTAC promoter and PT7 promoter. And the vector must have the multiple cloning site so that you can insert the gene of the enzyme that you want to express in E. coli into this multiple cloning site, and you can produce it in E. coli. Now, E. coli, even though it's a very useful bacteria for expression of the protein, but it's, if you know, the origin of the E. coli is from feces, human feces. So many people still think that is a dirty bacteria, and it's also sometimes some E. coli can be dangerous. It can make you diarrhea. Some enzyme can actually lead you to have a severe sepsis. All right, so E. coli is never a good bacteria if you want to use it for food applications. And when I want to make oligosaccharides, I want to apply oligosaccharide for food and feed. So using E. coli is not a very a pleasant expression system, even though there is one E. coli called K12 that is approved for food use, but it might be better to try a different expression system. Again, with the collaboration with Dietmar in Boku and Vincent in Norway, we developed a food-grade expression system to produce recombinant enzymes for food use, okay? The expression vector for making enzyme is called PCIP base, okay? This is also from another collaborator in Canada. So you'll see that when you are a scientist, you collaborate with people around the world, all right? And then what would be the bacteria that is food grade? You probably know very well because you are in food. So Lactobacillus plantarum is one of the very common food enzymes, food bacteria, right? This is for fermented food. So we decided to use the expression system called PCIP expression system to produce a recombinant protein in Lactobacillus plantarum. So we get this expression vector, we did engineering, and we cloned the genes for manganese and clone the gene of our enzyme into this expression vector. And I can come back and will tell you a little bit more about the food grade expression system. All right, now we come to look at the research in my laboratory, which related to this talk, which is I want to use value creation of agricultural waste using enzyme technology. All right, so, okay, this topic is a hot topic now. If you know that this day we are entering into circular and bioeconomy, okay? EU already said about the EU has launched a circular economy strategy because this world needs to go into this direction if we want to live in this world happily, okay? They want to closing the loop of product life cycle through greater recycling and reuse and bring benefits for both the environment and the economy. So this is a big issue in Europe that we have to use green technology and we have to use circular. We don't want to waste any waste anymore, okay? And we have to preserve our environment. At the moment, the environment is very happy because we stopped doing many things that hurt the environment. You heard about the ozone is closed and everything because nobody have industry that much going on with COVID. But then the recycling is still a problem. We used to throw a lot of waste, all right? But anyway, so the idea of EU is that we want to reduce, reuse, recycle, and we create energy and dispose, all right? And of course, this circular economy, one thing is that the enzyme can be very useful, okay? We have to have, with the circular economy, we have to have a solution for residual waste. And in this case, what our laboratory is dealing with is agricultural waste. So this is an example of a company called AgroCycle that they make everything circular, all right? So if you have animal feedstock, you get a biomass, all right, and then you can try to return many things. Okay, fertilizer, come back, everything, they want everything should be in a loop so that we don't have to throw everything away. Okay, that's the idea of a circular economy. So, and this is an example of the 8 million project to do everything in University College Dublin, that they want to have zero waste. They're going to reuse everything. Okay, it's also in Thailand. I think everywhere in everywhere in the world, we're talking about this strategy to make everything a bio-circular economy. All right, so it just fit me, I myself very well, because I've been working with enzyme for the past 20 years, and then now the policy of EU, the policy of Thailand, the policy of the world said we have to recycle. We have to use a green technology to do this, recycle. And biotechnology is fit perfectly well. And enzyme fit perfectly well into this bio-circular economy, because enzyme is a clean energy, a clean technology, use very few, less energy, because it enzymes, so it works at a mild temperature. Doesn't need a lot of temperature, and this doesn't secrete toxic waste. Okay, and now go back to Thailand. Okay, what is in Thailand? Thailand, it is certain part of Thailand. I show you before, it has next to the ocean, right? So you have nice beaches and it is pretty warm. All right, so what are the resources in Thailand that we have a lot? We have prawn. Okay, in the certain part of Thailand, we have a lot of shrimp and prawn farm. But Vietnam, I think Vietnam probably #1 now. Thailand used to be #1, and now we're #2. We produce a lot of prawn, and this is for export. All right, and then there is this prawn waste, lots of prawn waste. And according to the circular economy, we want to reuse this waste and make it more value. Don't throw it away. Okay, so this is one of my research topic, is to try to increase the value of this prawn waste using enzyme technology. The other raw material that I use in my laboratory is coconut. It just happened that coconut is, in Thailand, there's a lot of coconut. So I want to use the coconut as a good resource to generate something more value, because when people make coconut milk or coconut oil, there are a lot of coconut waste. It's called copra waste. We just throw away to landfill or give it. It's a low value feed for animals. So if I can try to use enzyme technology to increase the value of this waste, it would be nice. So that is the main topic of my research. Okay, so before I go into this, I want to point out again that these two, the biopolymer that I've been using as a raw material for enzymes conversion into oligosaccharide, are two kinds. First is chitin biopolymer. The second one is called mannan biopolymer, that found in coconut meat, but it's not only coconut, it found in konjac, locust bean gum, coffee bean, okay, and also palm seed as well. All right, so these are the biopolymer that subject to my study. Okay, these two has chitin and chitosan, and this one has mannan. That's a polymer. So now, how to create value from this biopolymer? Okay, we know that in prawn shell, or even in insect, they have this chitin. All right, chitin is very tough because it's have to make a skeleton. All right, it's worn. There's no way that it's gonna be soluble in water. It's very tough. And if you want to dissolve chitin, you have to add very, very strong hydrochloric acid. So it's not easy. So sometimes what people do is that this is chitin. If you look into detail, this is a polymer of a monomer called N-acetylglucosamine. All right, it is this structure of sugar with acetyl group, N-acetyl group here. So if we cleave off the acetyl group, okay, there are two ways to cleave. You can use a strong base, or you can use enzymes. There's an enzyme called chitin deacetylase, okay, and you will get this deacetylate chitin is called chitosan. Okay, so it's a deacetylate chitin. It's easier to use because it's more soluble, but it is soluble in acid. Here is soluble in extremely strong acid and strong high temperature. But here, when you have chitosan, it would be soluble in mild acid. Okay, but it's still not where it has many applications to use in many things. Can use. It has antibacterial activity. It can stimulate plant growth, and etc. etc. There's many research on that, but it's still very difficult to use because this is not easily. It does not dissolve in water. But if you hydrolyze it with enzymes, okay, this enzyme would have to break beta-1,4 glycosidic bond, which is here. This is the beta glycosidic bond. So enzyme that can do the beta-1,4 hydrolysis, you will get a chito-oligosaccharide. Oligosaccharide, you know, is a short chains of chitin or chitosan. You can have chito-oligosaccharide or chitosan oligosaccharide, just a small, okay? When it's smaller, you can have a different size, and when it's small, it can be soluble in water, and that would be more useful. So a lot of research is now focusing on to produce chito-oligosaccharide because it's soluble and it has so many benefits to health. There's so many research going on. It has anti-inflammatory, anti-cancer, it modulates immune, it can have antifungal activity, stimulate plants and everything, many things, okay? All right, so the enzyme that can cleave is that if it is chitin, you can cleave with enzyme chitinase. But if it is chitosanase, you can use it to cleave chitosan. All right, now the second raw material is mannan. Okay, as I said, mannan can come from many things, but what I focus is on mannan from coconut shell. You know, this is coconut meat, right? And when you squeeze out the coconut milk or coconut oil, and you left this mannan. Mannan is rich in monomer called mannose, but in also mannose and glucose or galactose together. Okay, so this would be linear mannan, only mannan, but this is glucomannan, glucose, and mannose together. So there is a mixture of linear mannan, glucomannan. All right, but if you cleave it with the enzymes, you will get manno-oligosaccharide or MOS, and that can be done, by the enzyme called beta-manganase. So in my laboratory, we are working with basically 4 enzymes: enzyme chitinase, cleave chitin; enzyme chitosanase, cleave chitosan; and enzyme beta-manganase will cleave 1,4 glycosidic bond on the mannan. All right, so how you use this enzyme to create. This is example of the value creation of chitin waste. So Monica was saying that the insect have chitin. You know, prawn is also insect. Okay, but people eat this insect for a long time. It's very delicious, but nobody eats the shell. Okay, they throw the shell away. All right, but then there's now, as I said, it's rich in chitin, and you can make chitosan out of chitin. But then the price of chitosan is still not good enough. So if I want to add more value, if you can make it chito-oligosaccharide, it could be more valuable. So there's a company called Marine Bioresource that they can extract chitin, and they also use the chemical reaction to make chitosan. These are all done by using strong acid and base. So they have a company doing this, and they keep sending this to around the world. right? But then the value is still not high enough. And this is when I collaborate with them. We're going to use the enzyme to create keto oligosaccharide, so to make it more useful for application in many things, and also increase the value. As I said, the chitosan itself is water insoluble, so it's still not perfect. All right. So now, how you can produce COS? This means keto oligosaccharide. There are mainly two ways. You can use enzymes, or you can use chemical synthesis. All right. But then, if you compare these two methods. Okay, enzyme synthesis, enzyme reaction are gentle and controlled. Enzyme is a protein, so it works at the temperature of the living organisms, like bacteria is in room temperature. So it is a more mild condition. If you compare to chemical synthesis, the reactions are harsh, complex, and difficult to control because it's very hard, right? And then also enzyme is easy to control because you can add enzyme, but chemical is not easy to control. And then the next is that the molecular weight distribution of COS can be controlled, and this is random. The chemical reaction make it random, but here you can control by choosing different enzymes. And my laboratory has been using specific enzyme for bacillus, and it's been working pretty well. So if you can control the degradation, you can control degree of polymerization. If you cut for a long time or cut for a short time, but here you cannot control, it's hard to control. I think what is important when you compare enzymes and chemical method is that enzyme is more environmental friendly, whereas a chemical is noxious chemical and harmful because all the reaction is done in a very harsh solvent. So this is enzyme happen in water, okay, and then at a room temperature as a very mild pH, so it's better. And then the enzyme is very specific, right? Whereas the chemical reaction is not very specific, right? So in general, we prefer to use enzymatic method to produce oligosaccharides. Now, let me show you the first example of using enzyme, and this is enzyme manganese. All right, so this enzyme, the second material that we use in the laboratory in addition to chitin is mannan. And this we collaborate with the company called Maprao Thai, and Maprao means coconut in Thailand. So this company produce virgin coconut oil. So they have a lot of waste from the coconut meat after they squeeze out of the oil, and it's still rich in mannan and it's pretty clean. So what they did now, they just throw away or sell it as animal feed, and animal doesn't like it that much anyway. So it's very low value. So it's my aim to help this company increase the value and will indirectly help the farmer. So I propose to use manganese enzyme, propose to use enzyme technology to create value to this waste from the coconut factory as well. All right. So I'm just gonna give, this is my the timeline that I've been working in my laboratory in the last 20-something years since I work in Thailand. So the first step, I have to start to do the cloning and expression of the hydrolytic enzyme. And this is in fact, when Dietmar came into my life, because when I came back from the United States, I was working on phage display and technology. So, but then the phage display technology is the same is a technology of expression of something in E. coli. So it's the same technique that can be applied to express the enzyme as well. So 25 years ago, still not many people are doing this. So I started to establish my laboratory to do the cloning expression of the enzymes with the help of Dietmar, because I'm not an enzyme expert. And then after that, because there was this technique about directed evolution by Francis Arnold, and they talk about the directed evolution technique. So I try to use this technique to improve the property of enzymes. So to suit for the industrial application, all right, and also for consumer acceptance. So because then later on I have to move from E. coli to the food grade expression system, all right. But then once you have the product, the problem is that the oligosaccharide can be very complex. But if you really want to use further study the product, you need to do in-depth characterization. And later on, this is when I have to start to collaborate with others because you want to analyze this product in detail. So once you know everything in detail, then you start to explore more about the function of this oligosaccharide. But before that, we have to try to produce it in a bigger scale. You know, when you study enzymes, hydrolysis of the material, the scale is 1 millimeter in a small test tube. And that is enough to understand the function of the enzyme, the property of the enzymes, and the product in the small scales. But that's not enough for industrial application. So later on, I have to move on into more of a larger scale of preparation. And finally, you want to study the function of this oligosaccharide. I have to say that my career has been going to number 1, 2, 3 already. So I'm in the number 4 or 5, and I think I have about 10 years before I will retire, and that's it. All right, so this is my life. So I will tell you a little, as I mentioned before, in the beginning, when I first finished my PhD and start my own laboratory in Thailand, I was establishing a system to produce the enzymes. And I mean, as mentioned before, we use E. coli because it's the most common system. And we have 2 vectors that we've been using very, very well in the laboratory. It's called P-flat base and PET base expression system. Okay. This is a review paper to show that we found that this is a very efficient production system to produce the enzymes. And here we said it's good for beta manganase, but actually it's other extracellular enzyme. This is for secretion out of the cells. Okay, so there are many ways to secrete. When you secrete the enzyme out, it's very easy to collect the enzymes. The first vector is PMY vector from E. coli, MY, you know, so we make this vector by ourselves. We have this, as I mentioned before, the multiple cloning site. It is for cloning the gene into it. And here you can see most of the time we clone the gene into, you know, HindD3XH01 equal R1. Once you clone the gene in, all right, and if you have to clone it so that it is in frame with this tag, you can see 10 histidine tag. These tags are important so that it's fused to your enzymes, and when you want to purify the enzyme, there is a affinity column chromatography that binds to this 6 histidine tag so that you can purify the enzyme nicely. Also, we have a MIG tag, as I mentioned before. It can bind very specifically to the antibody for detection as well. I like to point out here that when you have the gene, you have to have a stop codon. If you don't have the stop codon, you cannot produce the enzyme properly because it keeps going, right? And as I mentioned before, there's a promoter, and this is under the tag promoter. So this is an enzyme that I created in my own laboratory, and I've been using it for the past 20 years, right? And so this is for cloning and expression in E. coli, because E. coli is a very good bacteria host for produce recombinant and protein. So this is the first paper that I published since I established my laboratory. We have a good secretion system, and this system is perfect for expressing bacillus enzymes. Okay, so you can see the amylase, this nice clear zone, chitinase, amanase. All of these enzymes can be secreted out with a nice clear zone here. So it's good for screening and also good for purification later. All right. So since I mentioned to you before that the enzyme is secreted out. All right. When the enzyme is secreted out, there are how the enzyme is secreted out of the bacteria. There are two mechanisms. That is going through two different proteins on the cell membrane of the bacteria. First is called SEC, secretion system. The other one is called TAT, secretion system. With the SEC secretion system, first the protein is in a linear form. So it's go through this channel of, it's called SEC channel, like a snake slithering through this. And once it is outside in the periplasmic space of the bacteria or into the culture media in the environment, it starts to fold so that the enzyme can function properly. So this is a SEC secretion system. The other way that the enzyme go out is that they fold properly already, and then with the folding properly like this, is go through a TAT expression system. All right, there are two ways. The enzyme that I use in my laboratory going out of the E. coli through the secretory system, meaning that it go out like this and it will fold inside the periplasmic space. Okay. How to know which enzyme will go out of the cell this way or this way? It depends on the signal peptide. Okay. The protein, the enzyme itself, okay, normally the enzyme will connect with the signal peptide first. And once it go outside, the signal peptide is cleaved off, then you get only the full enzyme that function outside the cell. So if you look at the enzyme that I work in my laboratory, okay, we did some careful analysis and we show that it is cleaved here. Okay, the enzyme can feel this is a native. In Bacillus, this enzyme is also secreted out. This would be the signal peptide in Bacillus, but it can be used by E. coli. So E. coli can detect the signal peptide from Bacillus and secrete out as well. But you can also fuse the A, which is a secretion signal in E. coli, to the enzymes. And then the enzyme can secrete out. But of course, it is not perfect as you feel is to the natural secretion peptide. So if you fuse a Bacillus enzyme with the E. coli signal peptide or A signal peptide, when you secrete out and you do some analysis, you'll see that you can get a mixture of the product because it's different. Okay, this is E. coli, and because this whole protein is a Bacillus protein, I think E. coli may get confused a little bit. Anyway, so it is useful. So we've been in our laboratory, we used both of these, okay? But we know that the A signal peptide lead to heterogeneous secretion of Bacillus, but is secrete in a high level. So sometimes if you don't have to worry too much, it's okay to use A, even though in the front is a bit different. So we published a paper in Bioresource Technology about the expression and characterization of the enzyme chitinase from Bacillus licheniformis that is suitable for bioconversion of chitin waste. As I mentioned before, we are interested in producing an enzyme that can cleave chitin, and we found that Bacillus licheniformis is a very good source of enzymes. This Bacillus can grow at a high temperature, so it's perfect because sometimes you want the reaction to be at a little bit higher temperature than 37, so that you have less contamination. Okay. So we clone the enzyme. We have the gene. We have the Bacillus. We isolate the gene of the Bacillus, and we put into the E. coli, and we produce into E. coli. And this is the experiment to show that you have the protein produced. And you can do some staining and you can see the enzymes. And once you have the enzyme, you would look for the property. You can see that this enzyme can withstand the temperature until 50 degrees and it come down. Okay, but this is good enough because when you want to use this enzyme in a real industry, you would try to use about 40 to 50 degrees. You don't want to use too much because it's too expensive with the energy. You don't want to use too low because then you can get contamination by other bacteria. All right. But later on, we found out that enzyme chitinase that will cleave chitin. But if you remember, chitin is so difficult to degrade. So it would be better. So we decided if it's so hard to degrade chitin and so it would be difficult to do the reaction. Because when you do enzyme hydrolysis, the substrate has to be soluble. So we clone another enzyme called chitosanase. But then Bacillus licheniformis does not have the enzyme chitosanase. So we have to look for the enzyme from Bacillus subtilis. Turns out Bacillus subtilis has a very good enzyme gene for making chitosanase. So we clone the gene from Bacillus subtilis and put it into E. coli. Okay, so we put the gene of Bacillus called CSN 726 into vector PMY202, so that we have this vector. And when we clone in, we can put in E. coli and we can produce the enzymes. This is the seismogram to show that the enzyme is working because we add substrate and we stain. So if the enzyme can cleave, if we have a clear zone, all right? So as I mentioned before, this is chitosanase. Chitosanase will cleave chitosan, which is a deacetylated form of chitin. And now of course, we do the enzyme analysis and show that it is not as stable of chitinase. Okay, it will degrade at 40 degrees. But then this is good enough. This is optimum enzyme activity. It means it works very well at 50 degrees. However, it degrades very fast. This is without the substrate. But if it's an enzyme and buffer, it degrades very fast. But if you add substrate, it is more stable. So it would be okay to use the enzyme around 40 degrees. So in our assay, we did the reaction around at this temperature, or maybe even lower. The good thing is that chitosan, as I mentioned, soluble in mild acid, so it's easier to do the reaction. We also, so that is the enzyme chitinase and chitosanase, and now we have a good enzyme that we can use to produce. So that would be the research that we have done. And as I mentioned before, that we also working on coconut, and coconut is rich in copper mannan. So we need an enzyme to cleave mannan as well. And the enzyme that can cleave is called mannanase. Okay, but if you want to call the enzyme, if you're a real chemist, they have a systematic names to call the enzymes. And this enzyme mannanase must be called endo-1,4-B beta mannose. So basically, this is an enzyme that cleaves mannan. All right, it's GH26. This is a glycosyl hydrolase family 26. This is the systematic name. So I have one student with a postdoc published this work, and he would clone the gene from Bacillus licheniformis, put into E. coli, okay, and express it and analyze it. And we show that it is suitable for cleavage of mannan, okay, and it can secrete very well in E. coli. So we've been published a number of papers on this, all right? And next would be to really use this enzyme to produce oligosaccharide. The second part is how once you I will I will stop for 5 minutes and I'll come back to the second part, okay? Okay, I will continue with the second part now, second step now. So in the first step, to review, so I have two agricultural waste or biomaterial that I would like to increase the value using enzyme technology. The first is chitin and chitosan from shrimp and prawn. The second one is mannan from copra meal, locust bean gum, or coffee beans or palm kernel. All right, so we need an enzymes. So enzyme chitinase, chitosanase will cleave chitin and chitosan, and enzyme beta mannanase will cleave mannan. So we have established an expression system in E. coli to express enzyme chitinase, chitosanase, and beta mannanase from bacillus, either Bacillus licheniformis or Bacillus subtilis. Put it into the E. coli and let E. coli express, and we show that this enzyme has a property that is very appropriate for industrial application. Anyway, so even though it's kind of good already, because Francis Arnold was published many paper about directed evolution of enzymes, and it's quite interesting as a laboratory to try to use her technique to improve our own enzymes. And as I mentioned, we only tested on enzyme chitinase. We used DNA shuffling techniques, and we showed that we could improve the properties for fold at 3 pH 3 and 2-fold at pH 6. All right, so, but we didn't really continue with this directed evolution to improve our enzymes, because later on we found that our enzyme is good already, so we were not interested in putting a lot of energy onto this. All right, this is to show that the enzyme that we improve, we could really use her technique to improve the enzymes. is possible, all right? So it's good for teaching in a way. So in addition to improve the property, the kinetics, to make it faster, we also try to improve the expression system, as I mentioned before. E. coli is good, but it's not very well accepted by the consumer. So we shift into the food grade expression system, all right? And this is for consumer acceptance. What are food grade? Food grade expression system is a system that is approved for use in food and is safe, okay? Mainly it's a safety. What are the expression system for food applications of expression system of recombinant protein for food application? First is Bacillus subtilis, all right? This is a bacteria that has been using for a long time. It's proof that it's safe. You can eat it and you will be okay. For E. coli, as I mentioned before, even though E. coli, it comes from feces and it can be dangerous, but there is only one strain of E. coli that has been approved by US FDA for food application, and that is E. coli K12, okay? The other types of bacteria that is commonly approved for food application is lactic acid bacteria. Of course, this bacteria has been used in food applications for hundreds of years, right? To make cheese, to make all fermented food, and kimchi, fish sauce, all kind of things, all kind of fermented product. It make the food product sour, right? Because of the lactic acid produced. All right, and we know that there are many kinds of bacteria, but the well-known food grade expression system, the first one is called NICE system, and that is come from Lactobacillus lactis. The other one is PCIP-based system. Okay, as I said, this is from a group in Canada that we are collaborating with. There are good system for expression of the recombinant protein in Lactobacillus species. So I will tell you only the PCIP-based system because we've been using it in our research. Okay, the vector, the expression vector is called PCIP409 or PCIP609. Okay, and bacteria is Lactobacillus plantarum. This is the bacteria that is used in fermented food to make kimchi. All right. I think, all right, but many kind of food, all right. So let's talk a little bit about this Lactobacillus plantarum expression system. It's food grade bacteria. It has this GRAS status, meaning generally recognized as safe status. This status is important for in Europe to be sure that it's safe. It has that. What is nice about plantarum is that it has a probiotics activity. It's considered to be a probiotics bacteria. Probiotic bacteria mean the bacteria that have been shown they have a criteria for probiotics. Basically, these are bacteria that show beneficial effect to the host in the colon, right? It has no endotoxins, no inclusion body, so it's very easy to produce the enzyme. Simple cultivation, non-aerated. Okay, it's a facultative and aerobe. It means it doesn't need oxygen, a lot of oxygen, so it's easier. If you want to grow E. coli, you have to put a lot of air in it that can take energy. This is much easier. You just sit there, let it work. But there also a drawback. It's need a complex nutritional requirement, right? It's not only a simple LB media. And because it's a lactic acid bacteria, you know that it acidifying the culture, it make the pH reduce. So this is something important. You have to make sure you control the pH, all right? But anyway, so we start to work on lactic acid bacteria expression system. So we got this PCIP vector from Canada. So what we did is that we cloned gene of interest into this vector, all right? And then this vector, the original vector, has a ERMB for erythromycin resistant gene. So if the way it works is that if lactobacillus is not resistant, will die in the present erythromycin. So if you put the vector in Lactobacillus plantarum, it will survive in the agar plate with erythromycin. That's been the bacteria has this vector, which including it has your gene of interest. This is for the molecular cloning. You need to have a system to select only for the bacteria that has a vector to express your enzymes. Now, if we clone the genes into this, it still have antibiotic resistant gene, which is not acceptable for food grade. So if we really want to make it food grade, we have to convert it into some other marker that is not antibiotic resistant. Okay, so what my collaborator, which is the Dietmar's lab, did is that they convert erythromycin into alanine racemase. Okay, alanine racemase is an enzyme that will convert amino acid L-alanine to D-alanine. You know, the mirror image of the molecules that always have in L form and D form. Turns out the L form is a form that you find in nature, all right? But D form, okay? The D form nature does not have. The bacteria needs it to produce a part of the cell wall. And the way the bacteria did that, they had an enzyme alanine racemase to convert this racemase from L to D. And as I show in this picture, the D-alanine is required to form the complex structure of the cell wall for both gram-positive and gram-negative bacteria. So you see, it's written here, D-alanine is a component of the peptidoglycan, which is a structure of a bacterial cell wall. So if the cell don't have it, the cell will die. All right, so we decided to, in my collaborator, use this alanine racemase as a way to select the bacteria in itself. Meaning, if you grow so basically, what you do is that we have a PCIP-based expression vector. The original one, PCIP409, you can do the selection. Because we did the selection with erythromycin resistant gene. We clone the gene of the enzyme in between this cloning site. And we published a paper. In that paper, we would fuse this enzyme with a different signal peptide. So we want to see which one is most appropriate to produce a recombinant enzymes. And we also take out this erythromycin resistant gene and replace with alanine racemase gene to make it food grade. Okay, so these are the different. So you have because you have a different expression vector, you have to express it in a different bacteria. The vector with the erythromycin resistant gene will be expressed in Lactobacillus plantarum, WCFS1. Okay, this one is not resistant to erythromycin. All right. For the vector with alanine racemase, we would express in Lactobacillum TLG02. Okay, this is an oxotrope. It's a D-alanine oxotrope. It means it cannot produce D-alanine because we clip off the, I think they get rid of the alanine racemase gene. Okay, so if you grow in normal media without adding D-alanine, the bacteria will die. Okay, so unless the bacteria has this vector. Okay, so there's no antibiotic selection, only just normal media. All right. So this is a way to get rid to deal with not have to involve the antibiotic selection, but it's also not the selection is the cloning is more difficult because selection is not as strong as antibiotics. But anyway, we made this construct all right? And then we put the genes into this, we compare to at first, before you want to develop an expression system, you have to try, okay? We don't know what is best. We want the enzyme to secrete it out so that it's easier to collect, all right? We have a gene of chitosanase and manganase, and we fuse with the different signal peptide to compare. First is the A signal peptide. The second one is a native signal peptide from Bacillus. We put into this expression vector, and when we express and see what happens. So this is a work by two students. One is a master's student, the other is a PhD student to did this. Okay, so, and this would be the picture of the manganese. You can see that in this one, in supranatant, it means the enzyme is secreted out into the media and we spin down the cell. The left part is called supranatant. You have a lot of enzyme in the supranatant. It means it's secreted out very well. And if you can compare, this is the one with the native Bacillus signal peptide, and this is with the E. coli signal peptide. You can see that lactobacillus, like the native enzyme from the Bacillus, better than the E. coli. Okay, Bacillus and lactobacillus are gram-positive bacteria, so probably they like it more, so it is expressed more. As for the chitosanase, it's the same, all right? So the native signal peptide and supranatant, you have a nice expression compared to this. This is compared to the E. coli signal peptide. All right, so we managed to show that the enzyme can express very, very well. You can see these big bands of the enzyme secreted out in both supranatant and also, if in cell extract means certain part of the enzyme is still inside the bacteria, but that's okay. We are focusing on this in the supranatant because it's easier to purify. We also did the assay to look at about the cleavage, and we show that if it is a native signal peptide from Bacillus, is cleaved very nicely. Lactobacillus can detect Bacillus nicely and cleave very well. But this is the A signal peptide is miss the cleavage a little bit, but still okay. Chitosanase as well, right? So we have a nice system to produce the enzymes in Lactobacillus. But again, as I said, it's not good to use erythromycin, so we change into the alanine racemase. And also, once it's in the alanine racemase, okay, it can also express, even though in this picture it's not as good as erythromycin, but it also shows that we can express it very well. So both of these systems can be used to produce the enzyme for bioconversion of chitin, chitosan, or mannan later. We have only enzyme, though we have not gone into really use this enzyme to produce anything yet. This would require more funding, more research, more expertise in the biofermentation, bioprocess engineering. Anyway, we managed to publish this paper on the secondary production of the beta manganase from the Lactobacillus plantarum. Okay, we show a nice expression and everything. And next, this is again with Dietmar here, and then with another student from Dietmar lab. They did another assay when they anchor the enzymes onto the membrane. Now, in my paper, I secrete the enzyme outside, but in this group, instead of secrete the enzyme outside, they anchor it onto the cell wall or cell membrane of the bacteria. And this might be helpful in, you can keep recycle the enzymes. So they show that the enzyme can be recycled and it can be also used in the future. And this is food grade. So, but you know, we have not really gone to really use this enzyme for producing anything yet, but this is my last plan in my career, is to try to really use the enzyme to produce something useful, right? And in the beginning, I would just use the E. coli enzyme, the enzyme produced from E. coli, because it's easier. And of course, later on, I would move into the food grade expression system. And as I mentioned before, when you produce the oligosaccharide, it's important to do in-depth characterization. Otherwise, you cannot really understand anything if the product is impure. And in fact, there's so many people publication out there talking about the effect, about biological effect of phyto-oligosaccharides. But if you look into detail, the oligosaccharide product is not well characterized in the field, and it's very confusing because chitin chitosan is large. When you cleave into phyto-oligosaccharides, it has so many sides and it's mixture, and it's not clear. So it's very important when you go to buy this product. I mean, what kind of product is this? And when you compare different publication, it gets confusing because people use different phyto-oligosaccharide to do their research. So we think that it's very important to first really understand the characteristic of our product. So I begin to collaborate with Vincent Ising, okay, to ask him to help me with the characterization of our hydrolytic products. Okay, so we use the enzyme Bacillus subtilis chitosanase, and we cleave the phyto-oligosaccharides, and we use mass spectrometry, NMR. We use a size exclusion chromatography, which I won't go into detail, but we manage to understand the enzymatic activity of our enzyme, and we can look at the product in detail. We understand that enzyme from Bacillus subtilis chitosanase 46A, okay, this 46 is mean from glycosyl hydrolase family 46. It has we confirm that it has an attractive property for bioconversion of chitosan from chitin waste. This is confirmed the previous work. We show that the enzyme uses non-processive endomo. This is an enzyme, people, they know there are many ways the enzyme works, and this is a non-processive. It may cleave and come out, it's not staying on the substrate. But it can rapidly convert various chitosan into a unique product mixture, which is different from another enzymes. Okay, we know this is a combination of our product. We can see very clearly. And you can see a different enzymes show a different combination of the products. Okay, so it can be very important when you really want to study the property of the oligosaccharide. Okay, so as for beta-mannan, it's easier to study because the substrate is rich in mannan. So we also look at the structure of the product, and we found that the product looked like this. So I'm about to also publish the paper on the function of this. Okay, this is the structure of the product that we have. And also, it is important as well to produce the enzyme in a larger scale. I mentioned before that we used to produce the enzyme, study the enzyme in a test tube, only 1 milliliter. But if we really want to produce, my research is moving toward the pilot scale preparation. And we are about to publish this work. This is the work that is actually not even published yet. But we can use the enzymes in a larger scale. And this is we change from 1 milliliter scale to 100 milliliter scale. And it should be 1,000 or even 10,000 liter scale later. So in here, we can, in a larger scale, we can still also produce the oligosaccharides, and if you can see, you can vary the enzyme concentration. You can see different product you can look. And in this case, you look at the oligosaccharide by using a method called thin layer chromatography. I don't know if you know this technique. So basically, you have a layers, a medium here, and you spot the phyto-oligosaccharide here. and you put into a tank and let it migrate by capillary force because of the solvent here, and the oligosaccharide move up and separate according to the size. All right, the larger one is still stuck here. And from this you can see, and you have a standard, you will know the product that we have is dimer, trimer, tetramer here. Okay, and from this we can calculate the percent yield, and which happen to be 100%. So the enzyme is very efficient in cleaving the product. Okay, so you can calculate from this. And also you can see here it's clear. It means it is completely converted into the product. And if we use either 0.5 unit per mil or 1 unit per mil. And from this, we have already filed a patent to produce this oligosaccharide, and we are start to see the interesting biological activities of these oligosaccharides. As for cost, we can get 100% yield, but for mono oligosaccharides, we get a much lower yield. We got only 10% yield because the mannan, you see like this. Okay, when you start to digest it, it is still only 10% that we can convert. The rest is still stuck onto the coconut copra. But 10% is not bad. We can collect this 10% and we can freeze dry it, so we can manage to get this product, and we plan to use it to study biological activities more. Okay, so we are still working on this, and I think this would be the future where we can use the enzyme technology to produce more of this, bigger than the lab scale, going to be the bigger pilot scale or in industrial scale later. That would be my hope and my dream that would become eventually. So now, if you look at the function of the oligosaccharide, as has been shown to have so many activities. For example, antimicrobial activity, antioxidant activity, anti-inflammatory activity, anti-cancer, I mentioned immunomodulating, wound healing, tissue generation, hypocholesterolemia deficit. So preservative, prebiotics. There's so many activities in the literature. Many researchers all around the world are studying on these activities. But as I said, the cost product that is used to study is a bit confusing because different labs using different enzymes, using different method. Sometimes it's not enzyme, but other method to produce this oligosaccharide. And they show different activities, but it's hard to see. It's not easy to compare between lab to the lab, okay? But anyway, so in my own laboratory, we produce our product. We have the in-depth characterization, and we start to look for the activities. And of all the activity listed here, I pick anti-inflammatory activity, because I think inflammation is the key to many diseases. And if we have anything that can reduce inflammation, I think it's a very good start. For example, COVID-19. You know that people die from COVID-19 because of the inflammation in the lung. And the inflammation is because the immune response is not properly. If you have a good immune response, it will kill the virus nicely. But there are some people, old people or people with comorbidities, somehow the immune system don't kill the virus properly, but not attack the virus, but attack the lung tissue. So secrete cytokines and everything that attack the lung, making fluid accumulation. And that is called inflammation. So inflammation can lead to a severe problem in our body. And if we can find something, many things or anything that can reduce the inflammation is nice. Of course, the immune response is important, but too much is not good. Okay, so but anyway, I'm interested in the activity to reduce the inflammation. Now, how do you study anti-inflammatory activities? There are many ways. Okay, we can do it in animal. We cannot test it on human, but you can do it animal. But then it's very expensive, and my lab is not equipped with the animal facility yet. So we start with something in vitro. Okay, so with the in vitro, you have to get the cells that is in the immune system. And for anti-inflammation, one inflammation activity is one key cells in inflammation is macrophage. This is not even is an innate immune response. Okay, it's involved in both innate and adaptive immune response. Okay, macrophage is a cell that eats any pathogen, okay, and sends some cytokines to kill the pathogen, right? Now, somehow, so far in the literature, when they study anti-inflammation, they use a mouse cell, or mouse macrophage. But mouse and human macrophage are different, and my lab, we are interested, and of course we are interested in human macrophage. So the first thing, we have to develop an assay to study anti-inflammatory. So we use human macrophage. But then the macrophage is a cell that must be differentiated from a monocyte. Okay, so you have to have a way to differentiate monocyte into macrophage. And after that, you would add something that make it inflame. And in this case, scientists use a molecule called LPS or lipopolysaccharide. This is a molecule on the surface of the E. coli or some bacteria. It's a very strong protein that when it will stimulate the macrophage, okay? To release many inflammatory cytokines, including interleukin-1 beta, okay? Of course, this is important for the body to release the cytokines so that we can kill the bacteria. But if there are too much of this, this inflammation will hurt the host cells as well. So it's a basic assay to look for anything that can inhibit this secretion of the cytokines that may reduce inflammation. So what we did is that we first pre-treat the macrophage with oligosaccharide, somehow add this, and then we challenge with LPS and see what happens, okay? So you can see in this graph, which we are about to publish, is that if you don't pre-treat with cause or anything, all right, you will get inflammation, which meaning you will release interleukin-1 beta. But if you start to pre-treat with something like curcumin or oligosaccharide, okay, you will see the reduction in the secretion. In fact, this picture is supposed to be not only interleukin-1 beta, it should be interleukin-6 and TNF-alpha as well. You will see a reduction in the response, meaning that cause can prevent inflammation. So we see this, and we are about to publish this data. So it's getting exciting that this cause, which is soluble, can have this. So that means it might be useful for anti-inflammatory activity later. Okay, like, yeah, it's actually in this picture. You can see that it reduced the expression of interleukin-1 beta, interleukin-6, and TNF-alpha. These are all cytokines that can cause inflammation. So you can see that if we add increased amount of cause, we start to reduce the secretion, is meaning it can block the secretion, which is nice. And the way it blocks is as good as when dexamethasone blocked the secretion. So it's quite nice, you know, dexamethasone is a drug that can reduce the drug that is commonly used for inflammation. It's a steroid, okay? And in our laboratory, we did this kind of experiment. We do it in my own laboratory. We use ELISA. It's a technique that you can look for this secretion of these different cytokines. And ELISA, I was mentioned in the last lecture, that is a method that uses antibody to detect whatever you want. So that means in this case, you have an antibody that can detect interleukin 1 beta, interleukin 6, or TNF alpha, and you can look. So if there's a lot, it would have a bright color. If it less, it less color. So with this experiment, looking for the inhibition of the secretion of cytokines, we show that hydroxysaccharide has the interesting properties of reducing inflammation. All right, and I mentioned also that we produce mono-oligosaccharides using enzyme manganese to degrade mannan into shorter mannan, which is mono-oligosaccharide. We use the same techniques using this enzyme from both E. coli and also from lactobacillus. And this is a result of the oligosaccharide that produced, and we test it for the prebiotic activities, meaning the ability to stimulate the growth of the friendly or beneficial bacteria, which is probiotics. And we show here that it helped to improve the growth of many bacteria, like lactobacillus, Acidophilus pembroke, and all this. If you see this, the moss is blue, so you see it helped improve the growth when compared to another oligosaccharide. So this is a result to show that it has a prebiotics activity. Unfortunately, when we test the effect of this on the bad bacteria like Salmonella, we also show that it also increased the growth of Salmonella. All right. So, but then anyway, another oligosaccharide also increased the growth of Salmonella as well. So we have to be careful. If that too much, it might not be good. All right. So yeah. So anyway, so this is the result so far to show that we can produce oligosaccharide that has a beneficial effect. And now, if you look in this picture, I mentioned before that prebiotics is the enzyme that can help the growth specifically of the probiotics. And probiotics, these are the probiotics that we tested in our laboratory. And we show that these probiotics are the bacteria in the colon of human. And it apparently has so many important beneficial effects to us. For example, in this one, I show 7 of them. It helps protect against pathogens, including Salmonella, Clostridium. We show that if we have a good probiotic, you don't have diarrhea easily because it helps prevent against these pathogenic bacteria. It helps to produce vitamins, especially vitamin K and vitamin B12. It helps to improve the immune system, especially around the guts. It somehow stimulates this immune to be strong, and it's very, very important for you so that if you have good bacteria, some people have diarrhea all the time, some people don't. It could be because if they have a good bacteria to help make the immune system strong. Not only fighting bacteria, other pathogens like number 1, but it's also help improve the immune system around the colon by itself, or intestine in general, or the whole body in general. It also helps promote the intestinal angiogenesis, meaning the blood vessel that come to give food and oxygen to the intestine. These bacteria somehow stimulate this angiogenesis. So, when you have more blood to come to your intestine, it's more active and it can work more properly. And it also includes to promote this fat storage inside the colon. So it helps to help balance, so you don't have, it somehow help. There was research showing that it reduces the fat, bad lipid in the blood if you have a good bacteria. All right, it helps acid production. It helps to produce short-chain fatty acid. And short-chain fatty acid has been shown to be very important for the growth of the probiotics, and also for many, it's also important for the other immune system of the host as well. And the last one, which is quite interesting and quite unexpected is that the gut microbiome, the probiotics, is also affect and modulate the central nervous system. It go to the brain. So you have heard a lot about this gut-brain axis, that if you have a good gut, good bacteria in your guts, it makes your brain better. Okay, so that's why it could make me you smarter, think better, less depressed. There were people who were depressed, have a gut microbiome is pretty bad. So it can be important for the brain. Okay, there has a study to show Alzheimer people, kid with autism has a gut bacteria different from normal people. So the probiotic is very important. So the scientists really want to try to find prebiotics, so any molecules, any food that can serve as a good food for probiotics. And we show briefly that oligosaccharides supposed to be pretty good to help improve so many good probiotics bacteria, and this could be useful as a feed of food, and we think this is a very good way to help improve the value of mannose. All right. And we also show that this is a oligosaccharides doesn't have any cytotoxic effect. You know, we want to eat it, you want to make sure it won't hurt the cell. We show that it doesn't do anything with the human fibroblast, so it should be okay. All right, and yeah, so I want to show this is a finish of my talk now. So this lecture that I'm giving to you is mainly done with the help of Dietmar Hartwich, the professor here at Boku, you know him, and also this is Professor Vincent Eising in Norway, who has been helping me a lot with study of oligosaccharides and chitin. And my PhD student, I mean, all the work is not done by me, but my PhD student, one or two of them has been working in Dietmar labs, and two of them had gone to Vincent's lab to do the work. All right, and this is the picture of the people in my laboratory. Okay, I think she's the one who worked on chitosan oligosaccharide, and he's working on mannose oligosaccharide, and he also from Cambodia. I have a student from Cambodia, from Myanmar. I used to have students from Vietnam as well. All right, and this is the latest group. I mean, Magda was not here anymore, but these are the two Austrian students who visited my lab. Okay, and I thought Stefan and Ju, I don't remember her name anymore. Okay. Oh, we have one more. She's not in this picture. Anyway, I always have, she's from Russia. She's not from Austria. I always have students from Europe to come to visit my lab. Okay, so I finish my talk now. Are you still there? Is people still there?
Yes.
Yes, we are.
Okay. All right. I think I'm about to finish. You have any comments, question, or anything before I close this lecture?
No, I don't have a question. I just have a question. Is this the last lecture or next week?
No more lecture. This is the last lecture. I have only 2 lectures, and next week there is no lecture, and on 20th you will present, right? Yeah.
Okay. Thank you.
Anybody else has anything to say? Okay. If not, then Ala and Natalia, you will be presenting on May 20th, right?
Yes.
Okay. And Monica, you're still there, or?
Yes, I'm still here.
Okay. You don't have any more question or anything?
No, I'm fine.
Okay. All right. I think, I mean, you already give a presentation, so that's fine. So if you don't have anything, I will put this slide onto the Google Classroom, and I might have 1 or 2 questions that I want you to answer, and then you can get a grade. Okay? It's got to be simple.
Okay, thank you.
Okay. All right. Then have a good day, and Monica, please look at the assignments in the Google Classroom, which will be posted in maybe a week or two from now, so that you can get a grade. And then Ala and Natalia, I will see you in 2 weeks then.
All right.
Okay. All right. Bye-bye, everyone.
Okay. Bye.